today will be on the exam. And then as far as the exam goes, pretty comparable to what you've seen for the quizzes. Expect a few multiple choice questions, maybe a few fill in the blank, several application questions, et cetera, et cetera. I don't think there's anything technically you have to draw, but you may have to recognize some structures. That's a possibility. Fill in a diagram, some variation on that theme. All this stuff I've done in the past. It's written for an hour, so the class goes for an hour and 20 minutes. The exam will be written to last 60 minutes, okay? So plan for that as well. I think that's what I got. You all have any questions? Comments? Concerns? Yes?
So it's all the way up to SIROs, is that?
Yes, up through the, that's like, well, CRISPR, I think
Okay.
was the last slide.
Yeah.
Yep, up through CRISPR. Yes, Bonnie?
Are we allowed to bring notes?
Oh, the note card question. Sure, why not? Okay, so you can have one 3x5 note card, front and back. Has to be handwritten, no mechanical reproductions. Has to be something that you can read like with your own eyes, not using like a magnifying glass like this. Just use common sense. But yeah, you can use 3x5 note cards. Not a problem. Other questions? Some of you are like, really? Cell students got really pissed last spring when they finally asked them like no one's ever asked before, so just ask the question, no problem. Anything else?
I always said like fill in the blank, matching.
There might be some matching, might be some fill in the blank, multiple choice, short answer, etc. A little bit of everything, yeah. I mean, it's not real, so I don't make a secret of how I write my exams. My exams are written such that if you come to class, pay attention, i.e. you're conscious sitting here, you should be able to do some version of like a 70 to a 75. Then if you spend some time in class taking notes, really paying attention, maybe asking a question along the way, then you should be in that kind of B to A range. And then if you're spending time studying outside of class like we expect you to, you can put things together, etc., then you're probably looking at an A. And that's how I write them. So there will be some questions you're like, well, that was an easy one. That's my are you in class conscious question. And then there's others that you definitely have to think about it. That's what we do. Does that help?
Yeah.
Sweet.
Also, is the note card one-sided?
Two-sided.
Okay.
Yeah. Yeah, because then I gotta go around like who's got, yeah, I just don't do that. So it's two-sided. Anything else? Yeah.
You did say you were fine with me recording, right?
Yeah, I'm fine with
Okay.
recording. Yeah, no problem.
Just making sure.
Yeah. All right, then onward and upward. So we're starting our unit on translation, and I actually had a conversation with one in the office where we were talking about stuff like this idea that what we learned early in the semester gets applied later in the semester. And you've certainly have seen that or should have recognized that already, right? We're talking about RNA processing and the structure of RNA and how it gets processed and what those 2' hydroxyls are for splicing, et cetera. That goes back to like pretty much the first day of class when we were talking about nucleic acid structure. This unit, there's 2 units, 2 exams, quote, quote, that students tend to struggle the most with. This is one of them, okay? So the translation stuff isn't too bad, but we're gonna take what we learned with transcription, about to learn with translation, and we're gonna put it together to talk about how genes are regulated in both prokaryotic and eukaryotic cells. And you have to put so many different pieces together to understand that puzzle that's where things start to get a little bit tough, okay? So I want to encourage you to stay on top of things, particularly as we start getting through the translation into the gene regulation stuff in a week or so, and just do your study, do your due diligence, come in and ask questions, et cetera, et cetera. Let me help you with the understanding. I'm happy to do so, okay? And so to lead things off, I have a present for you. And it's kind of a funny little present. I like this one. It does illustrate a point that I've made several times as we've gone through the semester. The structures we're talking about are 3-dimensional structures in 3-dimensional space. And we're going to be learning how the process of translation works with respect to both prokaryotes and eukaryotes. And in this particular instance, what you see is the large and small subunit of a prokaryotic ribosome associating and then disassociating a elongation factor called the FG. And so what you see is it getting added and then taken away, added, taken away, added and taken away. And what it demonstrates is that there are structural changes in the ribosome as it is interacting with its elongation factor, which are going to help facilitate its movement across the messenger RNA and the synthesis of a polypeptide. These things aren't static images in the context of what you see in lecture, or doing your studying in your textbook, etc. Keep going. They're actual, again, 3-dimensional structures. So there's EFG coming in, 30 and 50 S ribosomal subunits labeled for you. And I don't know, it kind of looks to me like a dancing chicken. That's what I've already sort of thought of this video as, is a dancing chicken, so I call it dancing ribosome. But it's kind of cool, right? Like seeing these structural changes that are going to occur along the way. try to emphasize that point as we're going through this particular material. So before we get to actually discussing the mechanisms of translation, we'll talk a little bit about proteins first as well. We need to spend a little bit of time talking about how we sort of got to the point of understanding that what was in our genetic information actually was providing for the genetic information to make things like proteins, okay? And so there were a series of seminal experiments done by George Beadle and Edward Tatum. They were using a model organism called Neurospora to address this question, which is what is in, quote unquote, the genetic information that encodes for these things that we call proteins and or enzymes, right? This was done in 1942. We're still working out that DNA is the genetic material, but as discussed in previous units, we thought, okay, well, it's either DNA or protein. So we knew it was something to do with these chromosome things, as it were. And really, they were the first that was able to directly show a relationship between this sort of amorphous idea of what we call a gene and how those quote-unquote genes led to the production of proteins and specifically enzymes and metabolic pathways. And so as far as it works goes, or how it works with respect to the model system, it's remarkably similar to what we've already seen with respect to the yeast model system that you're working with in lab. So it's still a fungus. It's a cycle really, so we can kind of start anywhere. So let's just start up here with some spores. And just like you've seen in the context of Saccharomyces cerevisiae, Neurospora comes in two different mating types. You have an A mating type, or a lower A mating type, if you will, and a big A mating type. It's similar to A and alpha in the context of Cerevisiae. Each one of these individual haploid spores can germinate, and as they germinate, they start dividing, and they form this mycelial structure. Mycelia being these long, sort of fibrous connections of cells that are produced as the cells go through sort of repeated rounds of cell division. Both sides of this slide, they're doing the same thing. Eventually, a mat A, or a big A, if you will, and a little a mycelium can get close enough to one another where they'll sense the presence of each other, and it allows them to fuse to now form a diploid cell. But this diploid cell in this particular instance is going to be quote-unquote binucleate. In other words, the two individual nuclei are held separate away from one another. And then after a period of time, the nuclei form to form a sort of traditional diploid nucleus. Under the right conditions, this cell that is containing the diploid nucleus can go through rounds of meiosis. So here we are, go to our diploid cell, it's going to go through meiosis, first division. First division is, remember, when they become haploid, and then second division, and we now have our 4 haploid spores. Something that Neurospora does that Saccharomyces does not do is that those 4 haploid spores will each go through one round of mitosis and then mature into their final spore structure, ending up back at the top of the life cycle with haploid spores, and around the cycle goes. And so once again, just as we discussed with Saccharomyces cerevisiae, Neurospora has this ability to grow in a haploid state, but it also can do sexual reproduction, recombine the genetic material in the context of meiosis, so crossing over is occurring, independent assortments occurring, all that good stuff, to form new haploid cells. And so it was with this particular model system that Beadle and Tatum were working to again get at this particular question: do these things called genes actually code for enzymes? And so their experiment worked as follows. They took some Neurospora that was growing on complete medium, so this would be complete medium like we use in the lab here at AC, right? Basically a YPD version of the medium, which has everything that the Neurospora needs to grow. Exposed it to X-rays. We'll learn later at the end of the semester that X-rays are an example of a mutagen. They have the ability to cause double-strand breaks in the DNA, and those double-strand breaks can either lead, ultimately, if not repaired, to death, or in the process of repair, can cause mutation to accumulate within the genome. So now they have this mutated version of Neurospora. They take these Neurospora and they pull them out and again put them onto complete medium. So it doesn't seem like anything's different, right? But what they've done when they do this step is what's referred to as an outgrowth. In other words, they're making sure that whatever they mutated with their X-rays didn't completely destroy viability, but at the minimum allows the cells to grow in the context of this complete medium. After this, you take your Neurospora, you go back, take a small little patch of the mycelium, and you put it onto different types of minimal medium. And that minimal medium may have different components removed. And in this particular example, they put it in different types of medium, one of which I'll just show you in a second, lacks the amino acids for making protein, as opposed to other types of minimal media that might have some version of a different type of dropout. But at this point, from this particular tube, they now have on minimal media a lack of growth. So there's some mutation within the genome of this particular batch of Neurospora that doesn't allow that particular set of cells to grow on minimal media that lacks amino acids. So now what they can do is go back and take that same patch of Neurospora, here's the little line here, and put it back into a whole bunch of individual amino acid containing tubes, minimal media tubes, and they've dropped one individual version of, or excuse me, added back one individual amino acid to each one of the tubes. So here we add back glycine, we don't get a restoration of growth. Add back alanine, don't get a restoration of growth. It's not till, in this particular example, we add back arginine, now all of a sudden we get a restoration of growth. So the interpretation here would be that whatever was the original mutation, all the way back up here, whatever that mutation is, for some reason prevents the cells from making the amino acid arginine, so they have to have that amino acid added back to it. To figure out a little bit further what's going on with respect to arginine biosynthesis, they can then take that particular set of Neurospora and put it onto a variety of different media that have different supplements to it that include the compounds that are found in the precursor steps, if you will, in the arginine biosynthetic pathway. And so in this particular example, we've got our wild type cells. This is just a control. We don't add anything back. The wild type grows just fine. Add back ornithine, it grows. Add back citrulline, it grows. Add back arginine, it grows. So this is our control experiment. Wild type is growing under all conditions. We took the mutant cells, and we take the mutant cells, we put them onto minimal media, and once again, just like we saw on the prior slide, if you don't add back things necessary for the synthesis of arginine, the cells are going to die. But if we add back ornithine, they're able to grow. If we add back citrulline, we're able to grow. Add back arginine, of course, and they're able to grow. In the next row of tubes, they don't add amino acids to one tube, but in this second tube, they also didn't add ornithine. And so here we end up with, once again, no growth adding anything back that would allow for arginine biosynthesis, no growth if we don't add back ornithine, but if we add back citrulline, we restore growth, and of course, arginine, we restore growth. And then the last tube, same principle, no growth on media that lacks the intermediates, still can't grow in the absence of ornithine, still can't grow in the absence of citrulline, but if we add the final end product in the pathway, arginine, then the cells are able to grow. We take all of this information, put it in some version of a table like you see here, and it allows us to interpret the data and figure out for each individual group of mutants that you have, what individual step in the pathway has been disrupted due to the presence of the mutation. In other words, with our version of our strains that are unable to grow on anything that lacks a particular set of media, then whatever step is encoded that takes the precursor compound and converts it to ornithine must be the mutant step. As opposed to those, if we add back the ornithine and we're able to restore growth, well, compound A works just fine, or enzyme A is working just fine, but now at this point, gene B is the mutated version. In other words, just because there's ornithine present, if this gene isn't working, then we can't go to citrulline and ultimately to arginine. And then last but not least, the same principle with respect to gene C. In other words, if you have a mutation in gene C, it doesn't matter if the precursor compound is present, it doesn't matter if ornithine is present, and it doesn't matter if citrulline is present. If this is where the block is, the cell can't make arginine, it would die. It allows us to take this type of information and effectively identify the different individual steps that are present with respect to a biosynthetic pathway for a given amino acid, in this particular example, arginine. And so working through this kind of experiment, literally doing it again and again and again and again, interpreting results for all of the different amino acids. So they're working with literally hundreds, if not thousands of individual mutants, doing this same experiment again and again. You literally end up with something that looks like this, right? Like this is the biochemical pathways chart that you see in biochemistry. You may have seen it in your cell bio class, so I'll just bring it down to show you metabolism. Each one of these individual steps scattered across here is in some way, shape, or form going to be like occur through the process or the activity of a given enzyme. And in many instances, it's one gene that codes for the production of that enzyme. And in the context of this particular series of experiments, like you see here, it really led to what we thought of originally as the one gene, one enzyme hypothesis. This idea that there's something in the genome that encodes for an enzyme that does a step in a biochemical pathway. Now, we now know, of course, that that's not the case. There are examples of enzymes that require two different subunits from two different genes to make that final enzyme. And then we've also already seen in the context of this class how any one individual gene can have an alternative splicing arrangement, thus producing different proteins. And thus, more than one protein or one enzyme, in theory, can come from any one individual gene, okay? So lots and lots and lots and lots and lots and lots of work to basically figure out everything that you see in the context of this slide here, which of course you can't make that out, but if you go all the way through Biochem 2, you'll learn a good chunk of what you see on this particular slide. All right, before we get to any questions, yeah, about, ooh, lots of questions, also just go that way, or John first and then work way around.
Okay, I think, like, I'm a little bit confused on this. So is the idea of this saying that even if, like, looking at mutant 2, for example,
Okay, mutant 2.
even if your precursors aren't growing, as long as you put the correct enzyme in at some point,
After that step,
it'll keep going.
That's exactly right. So if you add the intermediate after the step that's mutated, then you can get correct growth. If you add the intermediate that's before the mutated step, then at that point, it doesn't make a difference because eventually you're going to hit that block. This morning when I was driving up from Anna, exit 53 going to Howe was blocked, okay? So it doesn't matter if I had left Anna at any given time, whenever it was, I got to exit 53, I don't need to stop there, I'm just making the point, you can't get off at that exit, it's busted. Same thing, if this particular enzyme, the gene that encodes it, so the DNA is mutated, you make a non-functional protein, doesn't matter what extra you add to the media. If this step is mutated, the cell can never make arginine and it would die. You have to get this would be your group 3, right? You would have to add back arginine directly to support viability of cells that have a mutation in this gene. As opposed to here, if you have a mutation in gene B, if you add back citrulline, the cells survive. Why? Because citrulline is produced from B. Doesn't matter if B is non-functional, citrulline's being added in the media, the cells still have functional C and they can produce arginine. So here you're adding the compound after the block and then same principle with respect to the ornithine. Okay, it's awesome.
So the 3 groups represent how many mutations there are?
Correct. So there's a whole bunch of background genetics that I'm not explaining intentionally, okay? But as they go through and do these experiments, you might envision that the first time they did the experiment, they get some patch and it's like, oh, okay, I got some cells that no longer are able to grow in the absence of arginine and I have to add it back. And then they take another sample. Oh, look, I got another tube where I have to have arginine to be able to get growth going, right? So now I have 2 separate independent isolates of cells. The question is the mutation in the same gene or in a different gene, right? And so that's what I'm not telling you. There's a way in the background, in the genetics, to basically go back and identify how many individual, quote, hits you have relative to your pathway, and then you know how many individual groups, if you will, that you have to work with. In this case, in this example, it's 3. Now, arginine biosynthetic pathway is actually relatively short. It's not an accident the textbook uses this example, because in the end, you really only have to have a few genes from precursor compound to get the synthesis of arginine. Some of the different individual amino acids in terms of their biosynthetic pathways, if you start from basic precursor, it could be 8, 10 different steps. The principle's the same, nothing's changed, it's just a more complicated example to try and visually look at. Okay, but they were able to understand that, like, hey, we have 3 individual groups. It's sort of, let me give you a different example. If you think back to that very first day in lab, we did the lab introduction, and I talked about how we can do genetic screens to identify things. And I gave you the example of Randy Sheckman. He was the one that went through and basically identified all of the different individual genes, i.e. proteins, that are responsible for protein secretion. And even though there were like 80 some odd genes he identified, they all fell down into like 5 different groups in terms of where they cause a block. And then within each of those groups, he had some that had the same mutation, others that had mutation in different genes. So then you only had to identify a subset of those because they cause the same phenotype, you know that you've hit that one gene multiple times. Does that kind of make sense a little bit?
Yeah.
Okay. Question was answered, awesome. Okay, any other questions about this? Yeah, it's hard to understand, like, how did we get to 3 groups? And that's, like, the genetics is super fascinating. We've done it in our lab, actually. The SBT4 mutant was actually one of 13 that we work with, is one of 13 individual isolates that we found that suppressed the PGM2 mutant, and we hit SBT4 twice in that group. So I've got others that I haven't identified yet. But I don't have to go back and screen both of my SBT4 strains to identify them. I identified one, they fall in the same complementation group, boom, I'm ready to go. So, all right, good times. Genetics, all kinds of fun stuff. Okay, so before we talk about translation itself, the actual process, we're gonna spend a little bit of time building ourselves up to the point that we can actually talk about translation and identify some features that are going to be necessary to understand protein synthesis. And of course, the basic is just beginning with what are proteins, right? And so proteins are, of course, a polymeric structure. The individual monomer for proteins is going to be the amino acid. So here's our basic structure of an amino acid, where you have an alpha carbon, an amino group, a carboxyl group, a hydrogen atom, and then some version of an R group or side chain that's going to be attached to that central alpha carbon that is going to identify which of the 20 amino acids this one actually is. So all amino acids are going to have this basic set of structures associated with them, and then you have the R group on the side there that is going to identify the individual amino acid. We said that proteins are going to be polymers, right? You take individual monomers, join them together, and the process of joining them together is what you learned in your prerequisite courses, which would be, of course, some version of a condensation or a dehydration synthesis reaction. Effectively joining the amino group to the carboxyl group of a previous amino acid, remove the water molecule, and you end up with this functionality that is unique to proteins, which is going to be an peptide bond. As far as the direction of synthesis goes, we're always going to take the incoming amino acid and attach it to a carboxyl group. So this is now amino acid 1, this is now amino acid 2, attach amino acid 2 to the carboxyl group of amino acid 1, remove the water molecule, and boom, we now have a dipeptide. If I wanted to attach a third amino acid, it has to be attached to this end. In other words, there's a polarity to the synthesis of a polypeptide chain, really not unlike in terms of what we saw with nucleic acid synthesis, right? Nucleic acid synthesis always goes 5' to 3'. In the case of polypeptide synthesis, it's always going to go from what we term the N terminus, the end of the polypeptide that has a free amino group, to the C terminus, the end of the polypeptide that has a free carboxyl group, okay? So a little bit of review of what these things look like. Probably a little bit different than your prerequisite courses as I show you the different types of amino acids. In this particular class, you have to know them all, okay? You don't have to be able to draw them per se, but you have to be able to identify and know the biophysical properties of all 20 of them, okay? So in this particular instance, yes, it's not that bad. You can do it, I promise, okay? I mean, my freshman, like, identified, what, 12 you had to know or something like that? It's like just straight learning. It's not too bad, okay? But it's important because later on, as we refer to proteins and like modification on a serine, if you don't remember the structure of serine, it's like, well, why does that get modified, etc. All right, so as far as the different groups go, the first group would be our non-polar aliphatic R groups. There's lots of different ways in which you can break these up. I like this particular version that I'm going to show you for a variety of different reasons. And so here we're looking at non-polar groups, and they're also some version of an aliphatic side chain. And it begins as the most simplest, which in this case is going to be glycine. Yes, it's just a single lonely hydrogen that is going to represent the R group. Don't be confused. The hydrogen that is common to all of the individual amino acids is still present on glycine. Its R group is just another hydrogen. And then from that point forward, we start building things up. Alanine has a methyl group. Valine has this branch structure. Leucine, isoleucine, etc. Building your way up. Okay? So all of these particular amino acids, as you see on the board, would generally speaking be referred to as non-polar with regard to their side chains. There's nothing really in any of these side chains that can form solid interactions with water molecules via things like hydrogen bonding. And so these particular amino acids are termed non-polar or hydrophobic, if you prefer that term. As far as the next group goes, we have polar amino acids. Notice that the side chains in each one of these instances, hydroxyl, hydroxyl, here you have some version of a sulfhydryl group, etc. All of these have the ability in some way, shape, or form to interact with aqueous solution quite readily. Okay, more or to a lesser degree, depends upon the amino acid. You'll learn about that in later courses. Just generally speaking for us, all of these are polar, and they are also uncharged at normal circumstances, if you will, in terms of what you find in terms of like environmental conditions of a cell, okay? So all of them have some ability to interact with water. They're polar or hydrophilic. The next group would be those that are also polar, but they carry, generally speaking, some version of a charge associated with them. So the charged amino acids come in two different groups. You have your acidic or positively charged R groups, and then, sorry, basic, and then your acidic or negatively charged R groups down here at the bottom. Each one of these individual side chains on the top is a positive charge associated with them, as opposed to the bottom group, it's a carboxylic acid in each one of these instances that are giving the negative charge. The proton would disassociate under normal circumstances in the context of a cell, okay? So each one of those that you see there. And then the last group back here on the bottom side, these are also going to be hydrophobic amino acids, but they have some version of an aromatic R group or an aromatic ring that's a portion of their R group. So just like the ones above them, they're going to be non-polar. However, they all have some version of a ring structure associated with them. Sometimes I get this question, well, why is tyrosine considered in this particular group? So the tyrosine and the hydroxyl that's present on here makes this end hydrophilic, if you will, but that big, huge planar ring that you see here also makes it slightly hydrophobic. So it's kind of a funny little amino acid, as the case may be. And so these guys, we kind of classify them separately according to this classification scheme, simply based on the fact that they have some version of an aromatic ring that's a part of their side chain, okay? As far as pulling these things together, once you've attached your amino acids together, we hopefully remember we can start to think about proteins and classify proteins at different levels of structure. The first and most basic level of structure, primary structure, is quite simply what is the order of the amino acids in the polypeptide chain? What is the identity of amino acid 1? What is the identity of amino acid 2? Amino acid 3, amino acid 4, etc., etc. You have already worked with primary structure. You did this the first week in lab when you did the gene discovery worksheet. Remember, you copied and pasted the polypeptide sequence for your protein, and you did a protein blast to identify similar proteins, right? So knowing what the primary structure is can be useful in many instances in the context of thinking about science. Then you also have different versions of secondary structure. In other words, if we take this polypeptide chain and we start to wrap it up upon itself, we end up with the next level of structure, which would be secondary structure. Secondary structures, the different examples I'm going to show you, are going to be organized and maintained by interactions between the backbone of the polypeptide chain. So typically hydrogen bonding that's going to occur between the backbone component of the polypeptide chain. The backbone being this here that I'm tracing with the pointer. So amino group, carbon-carbon, amino group, carbon-carbon, amino group, carbon-carbon, that's the backbone of our polypeptide chain. So hydrogen bonds between these different components can allow secondary structure to form. Take all the secondary structures that can form, fold it up into some version of a ball, and then you end up with tertiary structure. Tertiary structure can be the final form of a protein. If a protein is only composed of a single polypeptide chain, when it's folded up in its tertiary structure, it can now run around and do its job, and that's the highest level of structure that it will attain. So for example, the protein actin that makes microfilaments in the context of the cytosol of our cells, it folds up, it makes this final individual polypeptide that folds upon itself to its tertiary structure. That's the final active form of actin. As opposed to other proteins where multiple polypeptides come together and you have quaternary structure. Quaternary structure and tertiary structure would be held together through interactions with regards to the R groups that are present on the side chains of the amino acids. So both tertiary structure, one polypeptide folded up on itself, quaternary structure, multiple polypeptides interacting with one another, it's going to be interactions between the R groups that are holding these two final structures together. And for what it's worth, in this particular example here, they're showing you hemoglobin. So you have individual subunits of globin, each coordinating an individual heme component, all folded together to form this final quaternary structure of the hemoglobin molecule. So as far as different examples of secondary structure go, I said you'd see a couple of them. This one running down here is what's referred to as alpha helix. These over here are going to be different versions of beta sheet, where beta sheet is two runs of polypeptide running adjacent to one another, as opposed to alpha helix, where it's coiled like a spiral on itself. Fun fact, it was the alpha helix that was the first structure that was identified in the context of biological molecules through the use of X-ray crystallography. So this is one way in which we can get secondary structure. This is another way. Two different stretches of polypeptide chain running against one another. Now when we look at these, there's actually two different types of beta sheet that can form. That's what's referred to as anti-parallel beta sheet. We're going from N terminus to C terminus from right to left on the top, N terminus to C terminus from left to right on the bottom, as opposed to parallel beta pleated sheet. In the case of parallel beta sheet, notice that both of these are going N to C in the same direction with respect to how they're going to be oriented. There's additional properties that are associated with each of these in terms of how their hydrogen bonds are going to be organized. Are they all adjacent to one another at basically 90-degree angles, or are they kind of off kilter with respect to one another? How the different amino acids are going to be aligned with regard to their polarity. I think most of that you guys hit in biochem when you do protein structure and biochem. I'll save that for Jim and John. And then the last example of a secondary structure that I like y'all to be familiar with is this one, which is referred to as a beta turn. A beta turn is going to be specifically what you see here, where you have 4 individual amino acids. And so what they've done is they've got this central alpha carbon labeled here for you. So here's 1, 2, 3, and 4 that's held together in a turn by an individual hydrogen bond across the gap, if you will, after the turn has been made. I like you to know beta turn because in the context of something like a beta sheet, if you have a run going this direction and a run going this direction, how are these connected to one another? It seems like in earlier classes, all they tell you is like, we have alpha helix and we have beta sheet, and that's great, but if it's a single polypeptide, how is this thing ever linked to one another? It doesn't have to be a beta turn, but commonly it is a beta turn. So it's the last example of secondary structure that I like you to at least be somewhat familiar with. Questions about amino acids, questions about primary, secondary, tertiary, quaternary structure. Should be mostly a review, hopefully firing off a few neurons in your head being like, oh yeah, I kind of remember that from somewhere before. Yeah, Abby.
Can you say again what bonds hold together tertiary structure?
Okay, good question. So in the context of secondary structure, and it's not actually shown in this version of the image, but you see it here really well in the context of the beta sheet examples, that secondary structure is going to be hydrogen bonding that's occurring between components of the backbone, right? So here's a hydrogen bond that's forming between this oxygen that's a part of this carboxylic acid group, as opposed to down here, the hydrogen attached to this nitrogen is actually acting as a hydrogen bond donor, and boom, you have a hydrogen bond right here, okay? So secondary structure is being maintained by hydrogen bonds that are occurring between the backbone components, as opposed to tertiary and quaternary structure. The interactions are between some version of the R groups of the amino acids. And so the example that I like to give, because it's visual, hopefully it makes some sense. I've got polypeptide number 1, and I've got polypeptide number 2, and they interact with one another to be able to form a final version of protein that does job X, whatever that is. It's not too much of a stretch to say maybe we've got some version of a negatively charged amino acid there, aspartic acid, sure, why not? And then over here, we have this side chain of a positively charged amino acid. Let's call it lysine. Directly across here, those negative and positively charged amino acids can form a salt bridge and thus help hold these 2 separate polypeptides together. Same thing up here. Maybe we have some version of draw this right. Maybe we have some version of like a serine sitting on here, so there's a hydroxyl in this particular position. Maybe we've got a threonine coming over from this direction, so I'm not going to draw the whole thing, but we have some version of a hydroxyl, and then we can get a hydrogen bond across there. Once again, side chain interaction. That works for holding together the components of quaternary structure, the 2 individual polypeptides, but you also have the same thing within each polypeptide and its tertiary structure. That these bends that are being maintained in some way, shape, or form might be due I don't know, let's put a glycine here, we'll put an alanine here. Let's do a bunch of alanines, so it's nice and simple. Okay, so a bunch of hydrophobic alanines. In other words, we have this hydrophobic pocket that wants to associate with it itself, and it keeps the protein in that folded shape. It's still interactions regarding the side chains of the amino acids that are that are holding together tertiary, and then of course again quaternary. That good? Other questions? All right. So let's put this more into the context of genetics class then. How do we know what the cell uses in terms of the instructions to make a polypeptide? And so this particular chart, quote unquote, the genetic code, we've seen this going all the way back to high school, right? That as a messenger RNA is being read, we know that the messenger RNA is going to be read 3 bases at a time, or quote unquote, 1 individual codon at a time. So codon is 3 bases together in the context of a messenger RNA that can be interpreted by the ribosome to code for an individual amino acid. And so, for example, UUU would code for the amino acid phenylalanine, UCU will code for the amino acid serine, et cetera, that you see on the particular chart here. Other features of the chart are going to be that it has inherent to it a codon that typically is going to be used to start translation, and then some version of a codon that is typically going to be used to stop translation. And so understanding how we go from DNA to RNA to protein is pretty fundamental to understanding how a cell is going to work, right? We've already talked about this version of going from DNA to RNA, that's transcription, right? What we want to focus now on is how we go from messenger RNA to protein. How is it that this individual messenger RNA is going to be read 3 bases at a time to code for an individual polypeptide? As far as the genetic code goes, there's a lot of unique things about the genetic code that make it work like it's supposed to. We've already established that there's some version of quote unquote a start codon. AUG coding for methionine, AUG coding for methionine typically will be your first amino acid in a polypeptide chain. Let's go to the second, it reads it 3 at a time. CGA, we go over to the genetic code over here, we find C, and then we go down to G, and then we go across to A, the amino acid arginine, there's arginine here. And then same idea, we just did CGA, UAU is gonna be tyrosine, and then GCC coding for alanine, UAA, that's one of our stop codons. Notice in this particular example, I didn't write the word stop here. Stop is not an amino acid, okay? When you write out the polypeptide sequence, whether you use the 1-letter codes or the 3-letter codes, there's just, you'll see when we do translation termination, there's no amino acid there, it's just where the end is, okay? So keep that in the back of your mind as well. Stop is not an amino acid, that's an instruction to the ribosome to stop the process of translation. And so when we're thinking about the genetic code, like it's sort of this basic question. How do we know that the code is here in this messenger RNA, like reading it AUG, then CGA, then UAU, how do we know that's how the code is read, right? Couldn't it just as easily be AUG, UGC, GCG, etc., right? How do we know that it's being read in one series of discrete steps, one codon at a time? And that's where we go into our genetics, right? There's a series of experiments that have been done that tell us that the genetic code is a triplet code, that it's going to be read 3 bases at a time. I think before I start this experiment, or walking through this experiment, I think it's a good time for a break so you can stretch your legs, and then we'll come back to this and walk through it. Clear-headed is the case, okay?